We are delighted to present the inaugural Lauterbur lecture. I am Pablo Ross. I am the chair of the Department of Radiology at Case Western Reserve and University Hospital's Case Medical Center. Maybe I'll do this. Can you hear me well? And I think it's a big day for Case Western. It's a big day for MRI. And we have the luxury to start this inaugural lecture. My task is to introduce the concept of the lecture and a little bit tell you about Paul Lauterbur. So Paul grew up in Dayton, Ohio, not too far from here, so he's an Ohio product. His bachelor in physics was at Case Institute of Technology in 1951. Today, the CIT, the Case Institute of Technology, is part of Case Western Reserve University. Then he went to the Mellon Institute in Pittsburgh, where he started to work on the use of carbon-13 NMR. There was a war, Korean War. He was drafted in the U.S. Army, and I believe after being sent by mistake to a tank battalion, he was readdressed to be part of the U.S. Army Chemical Center, where they were basically doing chemical warfare preparations, which is a place, undisclosed location in Maryland, not too far from D.C. Finally returned to the Mellon Institute, and in 1962 obtained his PhD in chemistry. Then moved in 1962, after completing the PhD, as an associate professor at State University of New York at Stony Brook. And he was there until he decided to move. But before the move, he discovered something that he called zookmatography, the Greek for I excite. So I am delighted that the powers to be changed zookmatography. Because it would be hard to say, ma'am, you don't have to have a zookmatography of the kidney or the abdomen or whatever. So the first MR imaging paper was published in Nature as a proof of concept in 1973. And after a few more years at basically Stony Brook, he moved to the University of Illinois at Urbana-Champaign in 1965. And he received the Nobel Prize for his invention of MRI in 2003, becoming the fourth Nobel Prize in the imaging sciences after, of course, Röntgen, then Madame Curie, then Dr. Hounsfield, and finally Dr. Lauterbur. A lot of you are really interested. John Dawson, who I will introduce in a minute, who is here, is the author of these biographical memoirs from the National Academy of Sciences. And this is a very charming picture of Paul Lauterbur. I can imagine describing the excitement of the protons in zookmatography. But this is Paul Lauterbur at Case Western Reserve, and a little bit of history of Case Western Reserve as an MRI. In 1993, 10 years before the Nobel Prize, Paul came here, and maybe some of the more senior members of the audience may recognize where this is. That is certainly not in the current Department of Radiology, but we see here Paul lecturing in MRI to our department. So, Case Western and MRI, first of all, we're going to claim as being Paul Lauterbur's the inventor of MRI alma mater. But it is clear that UH and Case Western Reserve pioneered MRI clinically. For my generation radiologists, when MR appeared while I was in training, Cleveland was the mecca of early MRI. There is a history of this department of 35 years by now of innovation. Started with Dr. Alfidi, who was the chair at the time when MR appeared, Dr. Modek, who is now at the Cleveland Clinic, Dr. Dort, that is so kind to join us today, Dr. Lewin, who is at Hopkins, and of course followed the very perfectly high pathway by Drs. Griswold and Golani. We are blessed with a key collaboration with Siemens. Certainly the MR programs at Case Western and UH would have not been the same without Siemens collaboration. We are happy that Anne-Marie Gremmens is here. Terry Talbot, who couldn't be here, has also been instrumental in the last few years, but this started 35 years ago, so it's been a very rich collaboration for all involved. Parallel imaging, MR fingerprinting are some of the major accomplishments of our MR group, but also now, starting today, is the home of the Lauterbur lecture. A little bit more about MRI in Cleveland. In the 1980s, MRI became a $1 billion a year industry, and started in 1972 with the first zoomatography paper. By the 1980s, the whole body human imagers were produced, and many were produced here in Cleveland. Cleveland was at the MRI revolution forefront. In 1983, there were more MRI scanners in Cleveland than in all of Europe. And the two of the premier companies that led the development were Cleveland-based. When I was a resident, to have a Technicare MR was absolutely the rage. And this was, somebody tell me exactly what happened here, but this was the very first Technicare installed at UH that somehow was housed in the Cleveland Art Institute in 1982. I believe that somebody in the department recognizes this technologist. This was one of the incidents that obviously oxygen is picked up by the magnet, but at that time nobody had any idea of the many quirks that MR had. But this is a perfect slide. I believe Jeff sent me that. Jeff Dwork, these slides, but you see the evolution from 1981 to 1984, and then you can see what happened in 2010. And I want to almost finish with this classic picture. I don't think that John is here today, but this is Paul. This is the dean at that time, Dean Cherniak, Ted Castell, who passed away a few weeks ago, and John Hager, who's still around kicking. But again, before I talk about this is my last slide, I believe, but the Lauterbur lecture is important because Lauterbur was an alumnus here at the chemistry PhD, discovered MRI in 1972. In 1993, this is the summary Castell lecture, 10 years later, the Nobel Prize laureate, and most important for us, he was the teacher of Vikas Gulati. So we are delighted to have the first Lauterbur lecture today, and I'd like to introduce Vikas, who is actually going to introduce the inaugural lecturer for the Lauterbur lecture. So please, Vikas.
Thank you, Pablo. To be asked to introduce Alan Koretsky at the Lauterbur lecture is a huge, huge honor. Both names are absolutely legendary in MRI circles, and anyone who works in this field would be awed at this task. I hope I can do them some justice. I could probably talk about both names without any help from this piece of paper, but the sheer weight of their names would make me potentially forget my own name. And so I figured it was better for a change. My students know I never prepare written notes, and I did for this one. So since this is a Lauterbur lecture, I want to take a couple of minutes at the start to talk about Paul. I want to note that Dr. Joan Dawson and Dan Lauterbur are here today. Joan is an MRI scientist in her own right, a really well-acclaimed NMR and MRI scientist, a muscle physiologist, and they have been very kind to travel down from Michigan to join us for this lecture. And this is a huge honor for the lecture and for me personally, so thank you. And Joan recently completed a book. It's entitled Paul Lauterbur and the Invention of MRI, and it's published by the MIT University Press, so those of you who have an inclination, you may want to check it out if you have that interest. And as has been mentioned, Paul was the inventor of MRI and was awarded the 2003 Nobel Prize for Medicine or Physiology for this invention and the use of gradients to make pictures of stuff that is smaller than the wavelengths of the stuff that you're using to image it is insanely creative, and it's a beautiful invention. But this is the most visible piece of Paul's scientific life, but in no way is it complete. Before he was working in MRI, he was a very well-respected NMR scientist whose work in heteronuclear NMR is fundamental in chemistry. And after the invention of MRI, he made a number of major contributions to the development of the MRI technology, MRI contrast agents, MR microscopy, model-based image reconstructions, which are all the rage today. The list goes on and on. He had major contributions to our field that are felt throughout the field of MRI. He was much more than the list of scientific accomplishments. He was funny, kind, and I like to say he was a scientist. And what I mean by that is that he didn't turn science on and off. It was just the way he was. And one of my very favorite stories of this is when I was a very new grad student, and I said to him, some probe that I was using was not very good. And he got this very serious look in his eyes, and he said, "Good has no scientific meaning in and of itself." And he was very serious, and I got scared. And so I spent the whole summer making plots about how this probe wasn't responding in the way that I expected when I tried to drive it and to do what I was trying to do. And these plots I took back to Paul at the end of the summer, and I showed it to him, and he smiled at me and he said, "Now you can say the probe is not very good." He was letting me know that all in fact was pretty good between he and myself. But the story, it has a lesson here. He was trying to teach me a scientific lesson in a humorous way. And he was all about letting you do whatever you wanted in the lab. You could explore any crazy idea you wanted, but he wanted you to do the experiment seriously, plan the experiments well, and most of all, use your language very carefully. And so this was, this was Paul in a nutshell to me, the scientist. The story also shows, I think, a warmth and a humor that was part of Paul. And I really do think that he saw all of us in the lab as a family. And this is sort of exemplified by the fact that all of us, every single year that I was there, were invited to Thanksgiving dinner to Paul and Joan's house. And I don't know of very many people who would open their home to, 40-odd people in the lab for Thanksgiving dinner. I mean, this is the most sacred of American holidays, I think. So as Pablo mentioned, Paul was a Case grad, Case Institute of Technology, which merged with Western Reserve University to form what we know now as Case Western Reserve University. A lecture in his honor, I think, is long overdue at this place, and I want to thank Siemens Healthcare and Pablo Ross for making this possible. So, with that, now I switch to talking about Dr. Koretsky. So, Alan Koretsky, like Paul, is a name in MRI circles that really needs no introduction. Like Paul, hundreds and hundreds of scientific papers have been produced, and like Paul, he's far more than the sum of these papers. Early in my grad career in the early 90s, Paul said to me, when I was talking about one of his papers, Alan is one of the youngest and one of the best. And high praise, and clearly, therefore, Alan is a very appropriate choice for the Lauterbur lecture today. Dr. Koretsky received his bachelor's in chemistry from MIT, PhD in biophysical chemistry from UC Berkeley. He's presently the scientific director and senior investigator at the Laboratory of Functional Molecular Imaging at the National Institute of Neurological Disorders and Stroke. Before leaving for the NIH, he had been in academia and had been the Eberly Professor of Structural Biology and Chemistry at the Carnegie Mellon University. So, Alan Koretsky has led at least two major revolutions in MRI. He invented the technology we now know as arterial spin, he named it arterial spin labeling, ASL, and this is a non-contrast method, no gadolinium required for measuring perfusion. And is in and of itself an entire field in MR today. Entire sections of the ISMR annual meeting are dedicated to this. I remember reading the paper on ASL multiple times, and each time liking it just a little bit more. And it's one of those really complete papers where I, as a young scientist, said to myself, I wish I could be this good and write a paper like this someday. And so ASL touches deeply on neurofunctional imaging, the imaging of stroke, and now is extending into measuring perfusion in multiple other organs of the body, such as the kidney. His work has grown over the years and now extends well beyond ASL in the neurosciences. It touches on almost all fundamental neural imaging. Alan also helped pioneer a major revolution in MR by bringing concepts of molecular biology into design of MR contrast agents, a field that we now know as molecular imaging. This too is a huge area of MR, and Alan Koretsky is one of the core names, helping develop molecularly targeted contrast agents for use in MR imaging. These two fields, ASL and neural imaging and molecular imaging, they are so large and so different that people spend their entire careers working in one or the other. It is almost unfathomable that one person would have the knowledge and the bandwidth to span both, and do it at a level where he's known as an absolute leader in both fields. It is of little surprise that he was awarded the gold medal by the International Society of Magnetic Resonance in Medicine, ISMRM, in 2002. This is the home society for all of us who do MRI, and the gold medal is the highest honor bestowed by the ISMRM. Alan is also, I've never actually met him personally until just now, except for brief moments at ISMRM, but Alan is known in MR circles as a quintessential nice guy who carries no airs about his fame. And as I said, I grew up admiring his work from afar, and it is an immense pleasure to meet him, let alone introduce him here today. So please welcome Alan Koretsky to Case Western Reserve University and University.
Wow. Too much credit, Vikas, but thank you very much. This for me is a triple honor. I've not been to Cleveland or Case Western, and I hold the program in high regard. Dr. Roos, thank you for hosting, but all he said about the innovation and the impact from here is quite true. So it's really great to be here at Case. The honor one. Honor two, it's wonderful to see Joan and Dan. I had the great honor of being at the 2008 symposium in honor of Paul at the University of Illinois, where I first met Dan. Joan, I know a long time ago because we both liked to watch those phosphorus metabolites change. So Joan, it's really an honor that you two came, so thank you about that. And the third, it's the Lauterbur lecture. Unbelievable to have my name associated, especially at this first. And it was an incredible moment for me because Mark Griswold and Vikas ran me down at an ISMRM and said, we're doing the first Lauterbur lecture, would you come? And and I said, what do you mean? You're crazy, why me? And second, of course I'd come. And they acted like, oh, thank you, that I'd come. I mean, it's unbelievable. So thank you for the naivety. I can't imagine anyone would say no to the honor. So it's a special honor for me because I've been chasing Paul more or less my whole life. As it turns out, that is, Paul is this huge figure. And you'll see from the work I talk about, I still hear him constantly about the work. So almost everything I've done derives from Paul. Actually, and if you're young and you want some good clues for work, go read Joan's book, because Joan has put some of Paul's notebooks in that book. She's reproduced some of those, and it's amazing. We got very interested in susceptibility imaging at NIH to characterize tissue, and there it was in Paul's book. I should have read the textbook. In his lab book, susceptibility would be a nice way to make an image. It's unbelievable. Almost anything you can think of is there. So if you want a good idea for contrast, go read Joan's book. It's unbelievable. But anyway, Paul, as you heard, his career began at the Mellon Institute, and it's this wonderful old huge columns. It's a fabulous building in Pittsburgh, and I had the great honor of starting my lab in the Mellon Institute thanks to Chen Ho, and my scanner was actually in the basement around the corner from Paul's C13 NMR spectrometer. An amazing place for NMR axle-botherapy. It was a great honor for me to be working there. I also realized that every time Paul's been someplace, it fuses. That is, I didn't case Institute of Technology became Case Western. Carnegie Tech became Carnegie Mellon when the Mellon Institute transferred. That was Paul left before that too. So everywhere Paul leaves, they finally get the fact that people should work together, interdisciplinary, et cetera, something Paul stood for. Paul was the faculty at Stony Brook. I grew up in Queens, and had I listened to my mother, I probably would have taken chemistry from Paul, but I decided to leave town. And most importantly, Paul served on our advisory committee at the Pittsburgh NMR Center. We were funded as a regional resource from NIH, and I was a member of that. And that's where I really got to know Paul and quite well, and had been out to Illinois a time or two. I must have met, the honor's really special because both Vickers and Mark worked with Paul, and so somehow Paul has been transplanted here. It's wonderful. I knew that about Vickers. I don't think I knew that about Mark until this visit, so it's that's interesting. But anyway, I got to know Paul a lot, and Paul quite well, and brilliance. For sure, but generosity was remarkable. I mean, he was free with the ideas, it just flowed. And I remember vividly after we had started this arterial spin labeling trick, Paul comes and goes, "I'd like to start working on that. Is that okay?" So he asked permission. He goes, "I don't want to be a great bear and step on your toes." And I said, "Please step, leap, jump, wonderful." So it's a very generous, even for those of us who didn't work directly for him. Okay, so this title, I hadn't realized the Pope would be visiting. I was given this title, Molecular Imaging Quo Vadis, by Matthias Hahn in Cologne, and I've used it on special occasions since at Paul's Symposium and now here. I didn't know any Latin, a good Jewish boy from New York, but since the Pope is here, it's good. Quo Vadis, Latin for where are you going? And of course, Christians have adapted it because the story is Peter's getting the hell out of Rome because he's going to get crucified, and along the road there's Jesus come to him, and he goes, "Jesus, where are you going?" Quo Vadis, and Jesus says, "I'm going to Rome to get re-crucified." Wow. And Peter goes, "I better go back." And he does. And so this is a very long journey involved when you use the term Quo Vadis. And somewhere along the way, you will meet and return to the Father. And I think you'll see throughout, Paul is pervasive in what I'll say. So the journey we're all on, it sort of begins in 1973. The title of this paper, Image Formation by Induced Local Interactions, that's an interesting title, Examples Employing Nuclear Magnetic Resonance. So Paul knew he had invented not just MRI, he invented a whole new way of imaging. I think we now call this near-field imaging. If I were the Nobel Committee, he would have won the Nobel Prize right along with the atomic force microscopist. There's no doubt. And he knew it by the title. It's very impressive. My favorite figure in this paper is this figure 3, where he has 2 tubes of water, and one of them has manganese sulfate in it, and he turns up the power to saturate. The longer the T1, the easier to saturate. The shorter the T1, the harder to saturate. And so you can see one tube disappears, that's the water. The other tube stays, that's the manganese-containing tube. So in the same paper, not only did he teach us how to image water, but he also invented contrast agents right there. And the contrast agent he used was manganese. So there are many firsts from Lauterbur. Vickers told us that he invented MRI. He invented contrast agents, first to use iron oxide, for example, as a contrast agent. First 3D imaging, that you could do a 3D image. Spectroscopic imaging was there. Paul talked all the time about how we're going to get brain function, so I say he anticipated fMRI. The reduced case space is now called compressed sensing, and who knew what Paul was talking about when he first started talking about that. He's had a love of getting to high fields and dreamed of 12 Tesla, and we're futzing around with 11.7 Tesla, so we hope someday we'll be close to that dream. And he loved small detectors for very high resolution NMR. So throughout there's not a piece of the MR scanner that Paul hasn't influenced in an important way. And a lot of this, so I think the reduced case space has flourished. I'm not sure the microdetectors yet has flourished, but I'm sure they will. So there's just thing after thing that we're all working on and we're thinking about. It's really remarkable. So right away, I'd say MR was a molecular imaging tool, and Paul knew it. With the use of manganese. So there isn't a distinction between MRI and molecular imaging in my mind. And the killer app that really made MRI grow quickly was the detection of multiple sclerosis. And this is a case of an MS lesion. This is a case of GAD. Give GAD to someone with a multiple sclerosis lesion, and it enhances, and the reason it enhances is the brain has no blood-brain barrier. So I call this molecular imaging of the integrity of the blood-brain barrier. And since the invention of MRI, there's been a constant move to add function to MRI. I dated 1974, John will probably correct me, where a number of groups began to do spectroscopy to study metabolism in intact skeletal muscle. Water diffusion shows up in 1984. By the way, in that original Nature paper, Paul points out that you'll be able to do spectroscopic imaging. He points out that you'll be able to image diffusion. It's all there. It's unbelievable, last two paragraphs. 1984, water diffusion. This pointing out that iron oxide particles would be very potent MRI contrast. Maybe this is the birth of what we really mean by molecular imaging with MR. Simultaneously, in Paul's lab and Jack Lee's lab at UPenn, these iron oxides are very potent. And two uses in Jack's lab, Perry Renshaw used them to show that you might be able to see targeting of antibodies. And Paul was quite interested in cell tracking, using the contrast to follow cells, and I'll have a lot to say about that. And it keeps going. Let's measure blood volume and blood flow with a GAD bolus. And then 1990 is a great year where things erupt. Seiji Ogawa explains about blood oxygen level dependence, MR. Mike Mosley shows that diffusion detects stroke. Mike Mosley shows that anisotropy and diffusion can map white matter. This first paper about tracking transplanted cells was published in 1990. You know, if you want to read Paul, it's hard because the work, he didn't publish quickly like many people do today. And a lot of times there's a thesis or an abstract. And they were really doing this work much earlier than 1990. But there's the paper in 1990. We were interested in gene expression, also showed that in 1990. And the point of all of this is all of this contrast came in animals first. And when I talk to you good human imagers, I always feel small because almost all of our imaging is animal imaging, and I feel like I have to justify it. So there it is. It's good to do preclinical models, and not all has translated, but all will translate someday. So molecular imaging of the brain, the whole goal of molecular imaging is to measure everything you can in the, from the scanner, and in our case, the MRI scanner. Here's a list, particularly interested in how to measure synaptic strength and synaptic connections and cell migration. And that's what I'm going to focus on, how we measure these things in preclinical models. So one of the things that inspires us, you saw Dr. Ruth showed the beautiful change in image quality. You know, it keeps going. That's only a 2010 image. And it would be fun to see the gap in between. Paul loved and was a great supporter of going to higher fields. You know, in the history of MR, there's always been a discussion, low field better than high field. And Paul said, damn that discussion. Let's get to 4 Tesla and then 12 Tesla. I'm in that spirit as well. This is the third 7 Tesla in the world entering the NIH NMR Center. But magnets are good, but you also have to be good with detectors because, you know, you should got to build those detectors well. And this is a schematic of an 8-channel detector that my colleague Jeff Dwin had built. We're now up to 64 channels and still, I'd say, close to 50 to 80% gain in signal to noise still to be had with improved detectors on the brain. So we still have a way to go, and who knows where the field will end. And this is across the board. This is our 11.7 Tesla animal scanner entering the NMR center. And so almost all of the data I'll show you comes off this 11.7 Tesla animal. And you know, that's been why get a 7T. I had animal data at 7T for a long time. Joan had animal data at 7 Tesla for a long time. So if it works in animals, it'll work in humans. It's that simple. So we've been getting 11.7 data, so thus let's get to 11.7 in a human MR. And here's our 11.7 going into the building and was making beautiful images, and sadly it's quenched. So this is the 11.7 going out of the building for repair. So can't show you any data, but we're expecting it back hopefully in a few months, where we'll be working to get human brain images at 11.7 Tesla. It goes into the same spot as the 7 Tesla. The reason that got vacated is because we sort of de-risked the actively shielded 7T. This is the first actively shielded 7T that Magnix, maybe it was Agilent, or maybe it was Variant at the time, built, and that's coming into a normal 3T bay, and so that our steel shield needed a magnet, and there's the 11.7. So the higher field, the better detectors, and the images get spectacular. This is now a relatively old image from my colleague Jeff Dunn. It's weighted to see the magnetic susceptibility differences between tissues. Different tissues respond to magnetic fields differently, and at the high field you can begin to see wonderful tissue contrast. That's the whole brain, and I blow up this box. Resolution here I think was about 0.5 millimeter by a millimeter or so. So that blood vessels show up dark. We've known that for a long time. The interesting issue was the white matter shows contrast and the gray matter shows contrast in ways that I'm not sure we anticipated. And we even label the white matter bundle, so no DTI necessary. You just look and you can find white matter bundles. And there's all sorts of features, we're not sure what they are. For example, this is labeled a U fiber, dense white matter running from one region of a cortex to another, but maybe it's a vessel, we don't know yet. So there's interesting features that have not been assigned yet, but really spectacular resolution and spectacular contrast at the 7 Tesla. So we look forward to getting close to the dream of 12 Tesla that Tom Budinger and Paul Lauterbur had. And just to show the cortex again, if you run around the cortex, you can see very interesting contrast almost everywhere you look. I'll blow up a couple of places. This is the motor cortex right here, very dense in myelin or iron, and we're not precisely sure whether it's the myelin or the iron dominating this. But as you move into the somatosensory cortex, you can see this wonderful transition zone. Very important traditionally in histology to identify areas of the brain. And there's now the somatosensory cortex. So this is in vivo cytoarchitecture. Many people now are excited about that. And here's another cytoarchitectural feature known as the stripe of Gennari. Again, densely myelinated, high in iron, that defines layer 4 in the visual cortex. So these are now cytoarchitectural features that can be detected by MR. So where are we going? First, I'd say in higher field, better detectors, we have to increase sensitivity, and that's because PET brags. It's the molecular imaging technique. PET's got millimeter resolution and can detect about 1 million molecules. We're stuck MR with micromolar detection sensitivity of contrast agents, but we win on resolution 100 microns. That's about 10 to the 9th molecule, so we're still 1,000 molecules away from PET. So 10 micron resolution is when things will get really interesting. In the animals, most of the images I'll show you are somewhere around 50 microns, and in the humans, people are pressing on 300 or 400 microns. So just an order of magnitude, and this whole business is going to get it's interesting already, it's going to get extremely interesting. So what I'm going to tell you about is work we've been doing with 2 contrast agents, both invented by Paul, and this is where almost everything we do, there's a trace that Paul left behind. The first bit will be about how iron oxide allows detection of single cells and the tracking of single cells, and in particular we've been interested in endogenous neural progenitor cells. So translating imaging to humans, it's very difficult, so we avoid that by trying to learn new biology. So our work is geared toward learning new biology, and if it crosses that divide to humans, wonderful, but right now geared towards biology. And the second part will be about manganese-enhanced MRI, and here we've been trying to see if from the MR we can find out something about synaptic strength between 2 connections between 2 neurons. Okay, using iron oxide to detect cell migration is now a robust area, unbelievable. Chen Ho adopted Paul's idea first. I was down the hall, and the criticism was unbelievable. Most of it non-scientific, most of it just based on hunches, and it's been wonderful to watch this field really blossom. Many, many papers now. And most of that work is label cells ex vivo with enough iron oxide, put those cells into an animal and watch them. Only macrophages have been robustly labeled in vivo, that is, add the iron oxide and let the cells take it up. So the whole idea of using potent iron oxide contrast to track cells comes from Paul. Here's the 1985 paper that said ferromagnetic particles would be good for a contrast agent, and it was used to image liver cell, Kupffer cell uptake of the contrast agent. And here's the 1990 paper where neural stem cells, they were neural transplants in 1990, labeled with contrast and imaged in an animal. So again, this is a field that owes entirely to Paul. And the thing that Paul knew is if you put a iron oxide particle, this is this is simulated for a 4 micron bead, a magnetic field of about 200 Gauss. What's interesting is the field from that small particle extends out to 100 microns. So the fact that the magnetic field around this particle blooms, I think the Lauterbur rule in our lab is 50 times the size of the particle or so, but easy if you've got enough iron to detect out to 50 or 100 microns. And so that's the amplification that you get with iron oxide. And, most of the field, the whole rage is to get nano, make nano things. We decided you needed millions of nano. It would be easier to play with micron particles, and so we use big micron particles, readily commercially available, and get taken up quite well into cells. These are liver hepatocytes eating 1.6 micron cells, all the way up to 5.8 little bowling balls in the cell. And Eric Shapiro, and this follows on work that Steve Dodd and I and Chen Ho did, was able to clearly show that that's enough contrast to see single cells. And this is well accepted. I won't prove that to you. One of our favorite applications was with Kathy Kelly at the NIH, where we loaded up some skin cancer cells that metastasize to the brain, and we could find single cells, and as you followed them, you can see the tumor growing. So here's one, and there's later the tumor growing, and here's another, and there's the tumor, and that cell was very heavily labeled, and there's the tumor growing. And so this actually opens up the possibility of watching a single cell divide and grow into a tumor. And I'd say this type of work is gotten routine. That is, PET will tell you it can image cells. Many fluorescence will tell you it can image cells. As far as I know, in vivo, unless you have a microscope, the only radiological imaging technique that claims single cell detection is MR. And so that's exciting and where we continue to focus. Not just single cells, but you can detect single particles. So again, this is the work of Eric Shapiro a number of years ago, where he took a culture dish of hepatocytes and sprinkled a few particles on top. So here's a cell with a fluorescent particle living in the hepatocyte, and image that, and what you can see are spots, different scale here. You can see the contrast, loss of signal due to the magnetic field gradients. You can see the contrast, and you can see these wonderful dipolar patterns for those of you who like magnetic particles and magnetic fields. Because of the single particle, it just gives you a beautiful dipolar particle. So you can see single particles. That got us all excited, and I said, well, you better check. Paul probably did this experiment. Lo and behold, there it is in an abstract of the ISMRM in 1986. The Lauterbur group took a 5 micron iron oxide particle. So Eric was a little better, 1.8 or 1.6 microns, and showed that you can image single cells. He did that with 5 micron resolution. Eric did it with 50 microns. So we're still a factor of 10 off of Paul. Still a factor of 10 off of Paul. But anyway, that you can see a cell with a single particle said, we can label anything now. We don't have to be efficient in labeling. And the target we went after are new neurons that are born in your brain near the ventricle. Let's call this the subventricular zone. And here's an injection of a virus that makes the cells express green fluorescent protein. And these cells, they born here and they migrate along the rostral migratory stream, and they enter the olfactory bulb. And there's still no consensus on what these cells are for. And so this is a target. And so here's the data, again, still from Eric. James Sumner has taken over this project, and Nick Ornpathy is the postdoc whose work I'll show mostly to you today. Injects and a lot of particles we put into the ventricle, but you can see 2 weeks later there's this beautiful contrast coming out of the subventricular zone into the bulb and spreading out into the bulb. And you can follow that, so you can track this wave of migration. Here's right after injection of the particles, and a little time later you can see contrast coming out. 2 weeks later it's hitting the edge of the bulb, and by 3 weeks the contrast is fanning out into the bulb. It's just a couple of cases of that. So we can actually follow in vivo the migration of these cells and and them entering the bulb. These cells are pluripotent. They'll become neurons. They'll become oligodendrocytes. They'll die. And James Sumner did some quantification. About 35% of those spots were astrocytes. About 30% were neurons. About 30% were oligodendrocytes. We label about half of the cells, so we're not fully labeling all the cells. And about 5% of the cells labeled are microglia, which represent dead astrocytes, neurons, and oligodendrocytes that got gobbled up by the microglia. So, and a goal would be, sure, wish I had contrast to tell you that spot was an oligodendrocyte, that spot's a neuron, that spot's an astrocyte. We don't know how to do that yet, but there's a lot of clever molecular imaging going on here, so there's the challenge for you. And one of the easy experiments we did was just let's take an animal and give it a lot of odor and see if we can change the distribution of cells. And so this is an exaggerated result, not fair. Here's cell all around the olfactory bulb. And in an enriched environment, you can see more spots, boom, 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 boom. Thus, maybe more cells migrating into the bulb. And here's another example. And this is where you can see the anatomy. This is the mitral layer of the bulb. I think you can see the small contrast difference between the mitral layer. This was due to a little manganese. We'll talk about that later. But it's another Paul. We began to do cytoarchitectural experiments with manganese, and there's an abstract from Paul on how you can see olfactory bulb layers if you measure T1. So even the notion to measure cytoarchitecture, there it was. So that's what I mean. I keep chasing. That's like the tail. No matter what we do, there it is. Paul did it. But you have to hunt for those abstracts, no doubt about it. So here's a case where the odor has enriched, where we'd rarely find a cell. Now it's pretty easy to find a cell in that mitral layer. And this was interesting because this is a layer of the bulb where these cells were not thought to be that important, and it's where we see the largest changes. And here it is quantitated in different layers of the bulb, the cell number in control animals and animals that had been exposed to an odor. This was amyl acetate, the odor. Not much change in the external plexiform layer, but there's the mitral layer where things have doubled. And in general, when you go looking histologically in the mitral layer, what you find is the cells are all these neurons, these interneurons that these new neurons become, and they can find examples of cells with the particle. So here's one of the dopaminergic producing interneurons that have tyrosine hydroxylase. This cell actually picked up 2 particles, making it easier for us to see. So counting cells, and there's an effect on odor. To do biology with a factor of 2 difference in these animal models is a little troublesome. So before doing anything, we're trying to boost this effect. And the experiment to try and boost the effect was to induce plasticity in the bulb. It turns out your bulb is regenerating all the time. Your olfactory neurons will die. You'll produce new olfactory neurons. And that process is very sensitive to whether you're smelling things. And so the experiment that Nicorn does, and this is in collaboration with Leo Beluscio, is just to stuff up one side of the nose in 3-week-old animals. And since the animal can't smell, the bulb actually shrinks, which is not so good. It's best to look at the surface in the sort of CSF space. You can see it's enlarging on this side of the bulb and this side is smaller volumetrically than the open side. And then you pull out the plug and the bulb grows again. And so during this growth phase is where we were going to try and bias to see if we could change the distribution of cells. Instead of factor 2, we'd like a factor of 4 difference, something like that before doing experiments. And so this just shows what happened in the normal animals. The bulb is growing. And it's amazing. This olfactory bulb, I thought these are rats. 3 weeks of old. We all say by 7 weeks of age, animals are adults, but there it keeps growing. Takes till about 12 or 14 weeks before the bulb settles down. And what's happened when you plug the nose is it stops growing. Quite interesting. So the size difference is not because it's shrunk, it's just stopped growing. And the interesting thing is if you reopen, this was well known, this is if you reopen, the bulb grows back and tries to catch up to its normal size. And it's during this phase that Nicorn has measured what happens with these cells. And here's what happened. He's measured 2 things. He's measured the rate that they move out of the subventricular zone into the bulb, and he's measured the distribution, how many get into the bulb. So here's the rate. Control animals, inject the particles, 4 to 6 hours, hardly any contrast. A day later, contrast is coming out quite quite robustly. In the occluded case, again, early not much, and later they're migrating, but a smaller distance. And if you reopen, we make the same measurement. So this is on the same animal, then how fast are the cells migrating along the rostral migratory stream? No way to do this in vivo prior to this experiment. And this is what happened. The cells move along in the normal animals, and if you occlude, they slow down. And then if you reopen, they speed up just about to control level. So the plugging the nose has affected the cell migration, and that's interesting. How the nose lack of sensing odor talks back to these cells is a new area that we will get very interested in. And then he also measures the rate that the cells enter the bulb. So they will migrate to the center, and then they radially migrate out in the bulb and just count the cells. Again, we just very simple segment and count white spots after we segment. And so here's the cell number in the olfactory bulb as a function of time. Here's the normal animals, cells come into the bulb. And again, well known, they fewer and fewer as the animal grows up. If you occlude the nose, a few less cells. And what's interesting, if we normalize to the size of the bulb, so how many cells per volume, they normalize. So it looks like there's a difference here, but that difference is because the bulb is shrinking. The exciting result, though, is if you reopen, wow, there's an explosion of cells entering that bulb that's regrowing. So as the bulb regrows, it's taking these new neurons from the subventricular zone and being added at about twice the number from control. So that's an interesting period, and in the end we were sort of hoping, and we'll add the odor on now to see what happens. But this opens up the whole issue of whether the cells are required for the bulb to grow, and that's this experiment now. Heather Cameron at our place has a mouse, a rat, which is engineered so if you feed it thymidine, a thymidine kinase substrate, you will kill things. So it's a genetic ablation experiment. And so you can do this regrowth experiment after Naris occlusion with and without these new neurons. So you can ablate these new neurons and ask if the bulb grows. And this is a remarkable result, and one that's quite surprising to us. So in wild type, if you occlude the nose and then recover, the bulb, as I showed you, recovers size. In these animals where we've deleted these cells, the bulb grows pretty normally, but from the Naris occlusion experiment, they don't recover at all. So inhibiting these cells has inhibited the growth completely. And I'd say this is a surprise. I don't think anyone thinks these cells had so much control over bulb growth. And so this is going to keep us busy as well. So will any of this translate to humans? I think we're excited that we're counting cells in animals. The sensitivity is unbelievable. And there's been a handful of papers using iron oxide primarily to track dendritic cells in melanoma patients, in this case, neural stem cells in patients with brain trauma, in this case, bone marrow transplantation during spinal cord injury, in this case. And recently, Eric Ahrens, who likes to label cells with fluorinated agents, has shown with dendritic cells proof of principle that he maybe can follow cells. So I think the cell tracking, especially with the interest in all of the cell therapy that's coming, is going to grow in importance rapidly. And really all that's missing is the agent. The small nanoparticles that label cells well are off the market. The larger particles, Joe Frank at NIH has worked very hard to get FDA approval for labeling the cells, and he has a protocol, and it's got the IND. So I think there will be a burst of cell tracking in humans, and I suspect when we do cell transplantation in humans, it's going to be few cells. That's going to be interesting. It's going to really require the sensitivity of Paul's iron oxide. Okay, let me switch gears to the other contrast agent, manganese. As I told you, manganese was the first MRI contrast agent in figure 3 in Paul's paper. Maybe Joan can verify if I understand it right. I think Paul once told me that he submits the Nature paper, and back come the reviews, and one of the reviews said, well, why image water? Water's the same everywhere. What a stupid thing to image. And Paul needed to point out that the relaxation times were interesting and added figure 3 just for that reviewer. So listen to reviewers. It's not always bad. Sometimes they help you. Is that correct? I think that's, you don't remember, you don't know it that way. So it's not, it's my imagination. Like I said, Paul enters my imagination in many ways. But what's important about manganese, what's important about manganese and makes it so interesting to us and was interesting to Paul is it's essential. You're dead without manganese. So you eat manganese, you need a daily intake of manganese. Many enzymes require manganese as a cofactor, and so biology has evolved so that what you eat gets to everywhere you need it. And so manganese moves in interesting ways. It can enter excitable cells on voltage-gated calcium channels. So if you take a neuron and depolarize it, manganese will just come walking in. And the literature that uses fluorescent probes to measure calcium has used manganese as a surrogate. Manganese quenches those probes, so it's a surrogate for calcium transport. So it can tell you about activity. Once inside of neurons, the thing we're interested in, it will follow neuronal pathways and quite faithfully. And of course, manganese is a wonderful T1 contrast agent. That's why Paul pulled it off the shelf to make his figure 3. And so again, 1978, he follows up on the notion that you could use manganese as a contrast agent, injects it into some dogs and chops up the heart and shows that the heart will uptake manganese. And here's sort of the modern version of that experiment from Tom Hu and Andrew Arai, where they have a dog with ischemia. Manganese is infused. The normal tissue takes it up because it's fine, it's excitable. Manganese can enter on these calcium channels, but not in the ischemic zone. So that's the intriguing thing. And through the years, 3 nice ways we've used manganese in these preclinical models. Number 1, we can make activity. So Yi-Jen and Lin started this work, and there's the faux-pas stimulation as marked by manganese. We've not done much, but many other people have followed this sort of work up. If you give manganese just systemically, it gets into the brain and it settles to give you beautiful, stunning cytoarchitecture. It's this image that really stimulated my interest in using MR to measure cytoarchitecture. So there's an olfactory bulb, and you can see the olfactory nerve layer, the glomerular layer, the external plexiform layer. That's that mitral cell layer that the cells were on right there. So it really just gives beautiful contrast for cytoarchitecture. Most importantly for us lately is the fact that when you put manganese at a site in the brain, it gets taken up into neurons, and we think that's due to voltage-gated calcium channels. It gets packaged in those neurons, we think, into vesicles that then transport along microtubules to the presynaptic side of a neuron, where it can be released and reuptake. So it moves, that's the way the information flows. It moves in an anterograde direction, the neuroscientists would say, and it can cross synapses. And Robia Potler first showed that in a mouse, where she just put some manganese chloride in the nose, and there you can see it uptake into the bulb, and out of the bulb, you need to cross a synapse to get out of the bulb to the piriform cortex, the olfactory processing cortex, in this case in a mouse. So just to show the cytoarchitecture is just beautiful everywhere we look. This is the hippocampus, where the granule cell layer and the pyramidal cell layer are beautifully enhanced, but even layers in the cortex as well. And so the cytoarchitecture, instead of making a blob of a hippocampus, we can actually divide the hippocampus into its relevant cytoarchitectural elements. And we think the manganese distribution continues to be interesting at 50 micron resolution in the olfactory bulb. That first synapse from your olfactory neurons to these mitral cells is made in the glomerular layer, and they form little balls. And so we have assigned, Kai Shang has done this experiment and has assigned this contrast to specific glomeruli in the bulb. So these are now not just laminar information, but substructure within the laminae that we think we're seeing. really beautiful. So back to the tracing experiment. This says it's quite easy. Manganese chloride, no patent necessary, no company necessary. Just call up Sigma and put it in water and inject it. And so it's been quite easy, and many people have reproduced it. This is Nikos Logothetis, who tracked striatal basal ganglia circuits in the monkey. He showed that it moved just the way a normal tracer would move. And maybe the nicest work, I think, is Anne van der Linden, from Belgium, where she has used the manganese to track songbirds' neural systems. I don't know much about songbird anatomy, but you put it there and it traces to these two areas, and they just jump out at you as beautiful areas. The songbird system is very plastic, so it changes depending on hormones and depending on season. And Anne has shown that the amount that moves reflects the strength of the connections. And that's interesting, and what we've been very interested in recently. And, so this is work of Jason Tusaroni. If you really want to talk about strength of synapses, you want to be at this laminar area. And Jason showed that if we inject manganese into the thalamus, if you time it just right, you can watch the manganese entering layer 4 of somatosensory cortex. So here's layer 4. Most of the projections from thalamus enter layer 4 of the somatosensory cortex. And so this is where it's crossing the first synapse between thalamus and cortical processing. And so it's this kind of resolution and specificity that we'd like to address the question of where synapses change their weights. So this is this whole issue of plasticity. You're all learning something, or you're sitting there going, what the hell is he talking about? You'll still remember me one way or another. Or if you're damaged, for example, if you're blind, as these people are because of retinitis pigmentosa, your visual cortex is used by your somatosensory. Your fingers will all of a sudden start activating the visual cortex. So injury will just rewire your brain in really dramatic ways. A tumor in your language center will cause relateralization of language. So you'll be able to, the tumor, you'll be able to talk again, but you'll find that your language has moved to the other side of the brain. So the huge plasticity can occur due to injury. And so we're sort of interested in, the only model we have is this is happening because connections between neurons are changing somewhere. And we'd love to be able to tell you where and how and why. So this is an interest for MRI to detect plasticity. The functional and resting state connectivity literature is filled with stories where connection weights have changed. And there's a lot of evidence that your brain, the gray matter will get a little thicker, or white matter will get a little bit more myelinated with plasticity. And that's nice. It might be right, showing relationship between areas, but the synapses are at specific places. So instead of these gross aerial measurements, we'd like to be able to say, boom, right there is where the synapse is changing. And this is an extremely hard experiment. This can be done electrophysiologically in a slice, but to do it across a whole brain is there's no way to do it. And that's sort of that's sort of the goal. And so we make this injury model, which is just to sever the infraorbital nerve, which carries information from the whiskers. And we wait 2 weeks after severing that. We ask if there's been any change along the good whisker pathway. And of course, the best way to watch a change in a whole brain is fMRI. And so here's the fMRI data in an animal where we use the forepaw as a control. And so there's the forepaw activation across 3 slices. And here's the barrel cortex, and we're stimulating the good whiskers. This is the good whisker side. And here's the control animal in the barrel. And I think you can see, and I made fun of colored scale, so the students, excuse me, I've made this in a color scale. I promised you'd see it in gray too. It's easier to see, anyway, you can see there's been this big upregulation along the good pathway, and almost undetectable on the wrong side of the brain now becomes easily detectable. So the good whiskers are talking to the wrong side of the brain, and they've gotten strengthened on the right side of the brain. So this is the model, and what I want to tell you is where are the synapses changing. And so we revert to manganese. Shin-Yu did these experiments. My Shin-Yu, not your Shin-Yu. You have a Shin-Yu in engineering. I had a Shin-Yu now at the Max Planck, so not mine anymore. Inject a little manganese into the thalamus and watch the manganese move into the barrel cortex and the forepaw, so we can hit both. And with the infraorbital nerve, there's a little bit more that moves into the barrel cortex, and that's quantitated here. You can see there's about a 30% increase in the amount of manganese that gets from the thalamus to layer 4. And so we go, more manganese, a stronger synapse. And I go see my colleague, John Isaac, who loves thalamocortical connections, and he says, impossible, can't happen. I go, what do you mean, John? It's my MRI, we trust it. He goes, it can't happen because that synapse can't strengthen. All right, I said, that's crazy, every synapse can change. He goes, not that one. That has a well-defined critical period. Okay, so an animal is born, and for 2 weeks you can get plasticity at that synapse, but after 2 weeks, no more. No more. And he says that because of a slice experiment where they try and do this thing called long-term potentiation. That's the mark of plasticity, that you can increase the strength acutely in the slice, and can't do it. And I go back to the literature and I say, but John, look, sometimes there's a little more neural connections in the anatomy or a little less into barrel 4. He goes, anatomy, they don't look at everything, they don't know how to count, the image processing wasn't as good as it should be, as it is here. He doesn't like that, although there is anatomical evidence. And there was evidence from whole brains that you could strengthen this connection from the thalamus to the cortex as well. But when people looked in slice, they didn't find it. So it was in the whole brain, but not in the slice. Hard to know what it was coming from. So a good postdoc in John's lab, Sung-Soo Chung, it was interested in injury-induced plasticity. He took on the challenge of asking in a slice what happened. And so it's just a slice that includes the thalamus and the somatosensory cortex. Put an electrode into this little whisker barrel. There's one of that's a layer 4 cortical barrel. This we have not seen, although there is MR evidence that you could see these, and make electrical measurements there. And I don't want to go into much detail because this is an imaging talk, not an electrophysiology talk. But in the slice, the synapse has strengthened. That's what the connection has strengthened. It turns out that the synapse is stronger. So if you do a patch clamp and measure these miniature EPSCs, as they're called, so individual synaptic events, you can see right here that they're stronger, and that's shown here. About 30% stronger. Our fMRI is about a 70% effect. And then another experiment they like to do, which is to ask for more or stronger. And this minimal stimulation experiment, you can see, so you just stimulate so you get a action potential every other time, 50% of the time. You can see there's much more current when you do that in the animals that have had this plasticity. So we think each synapse is stronger and there's more of them. And most remarkably, this is the result that caused John to say it can't be a plastic synapse. This is an LTP experiment in the control animals where you don't see any plasticity, no long-term potentiation. Here's the average of a few animals. And now, in this slice, 2 weeks after we cut peripherally a central synapse, all of a sudden the plasticity has evoked. So this synapse feels young again. That's what we like to think, this induction of LTP. It's quite interesting, and I'm hoping quickly that we can find out enough about this so that I can make my synapses feel young again someday. But this much like counting the cells, this is from the MR, we said something's happened there, and the legitimate technique, the cell electrophysiology, says, yes, something's happened, and it's quite interesting. This synapse has reawakened LTP. Now, what about getting manganese out of the animal world into the human world? And I'd say there's two things that we're interested in, just because no one else is doing it. Number one, there's a PET isotopes of manganese. Manganese 52 has a 5-day half-life, so long enough to put into a brain and watch it trace. And this is the work of Galit Saar with Peter Herskovitz. The NIH cyclotron can make the manganese 52. There's a shorter half-life manganese as well, if you want to do the anatomical sort of things. And here she just puts some in the nose of the monkey, Robia's experiment again, and on day 1 you can see some in the olfactory bulb and the lateral olfactory tract and moving towards the piriform cortex. And on day 2 you can see it's out of the piriform cortex to the amygdala complex, really shown well on day 6. There's the amygdala complex. And if you look on day 1, not much in the prefrontal cortex, but by day 6 you can see some in the prefrontal cortex. So the manganese is moving through the monkey olfactory pathway as it should, and the PET can detect it. And 5-day half-life, you go, ooh, but remember iodine has a 5-day half-life as well. So it's quite interesting. We think the PET could move in the right clinical environment to this. The other way to translate to humans is to use this contrast agent that was FDA approved called Tesla scan manganese DP. It was shown a long time ago that it falls apart. It's really a release form of manganese, and many groups in the heart, and especially Per Junge, has used manganese DPP to enhance the heart. And we're back to asking about brain enhancement, and this just shows the pituitary loves manganese in the animals, and at the approved dose you can see enhancement in the pituitary quite nicely. And so we're after a couple of pituitary tumors that are radiologically silent to see if the manganese will enhance them. And manganese DPP is off the market, but it's still there, and we have it, and we've reformulated it, so we're giving it. It was on bottle for liver metastasis and never shown to be more effective than anything else, so it never caught on. But we think there's a potential for humans. So let me conclude, if you don't mind. I'll go a couple more minutes. Ficus's introduction of me was much too long, so I should get a couple of more minutes and show you another paper of Paul's that's really haunted me. Now we're all the way into 1993. As soon as Paul knew about iron oxide particles, he knew the distribution of those particles would change their MR properties. And what he did is he put particles into a gel, and the gel, he was very interested in gels, these smart gels that respond to their environment. And it's always been, every time you met Paul, he was interested in something. I don't know, it was like the early 80s, he was telling me about magnetotactic bacteria and how someone should go get that magnetosome, and I should have started working on it. Finally, I think in California there's a lot of work trying to figure out what makes those magnetosomes, as they're called. So Paul was very interested in these gels that will expand and contract for lots of reasons. So he put the particles in the gel and measured voltage here. So the gel was sensitive to a voltage. And so you can see the gel, the relaxation changing as the potential was changing across the membrane. These gels are pH sensitive. They can be sensitive to single molecules. And this work, I think, inspired Ralph Weisleiter and Alan Jasanoff to sort of engineer particles so that they would come together and fall apart. And this is Alan Jasanoff's work, where he put linkers that respond to calcium to aggregate the particles. And this has always been an attractive idea to me. Controlling aggregation and disaggregation is a tough problem, especially in a cell environment. And I don't think this has translated much yet, even into the animal side. So what we decided to do was to try and think about how to take that Paul's experiment, which is an incoherent experiment, that is, the particles affect relaxation of water in an incoherent way, and see if we can make it coherent. And Gary Szabo had the nice idea of, well, why don't we microfabricate? Well, John Moreland and I said, let's microfabricate something, and Gary said, let's microfabricate a magnet. So John's at NIST. NIST has wonderful microfabrication, and Gary began to make shapes like this. So he's got, this is nickel with a spacer and another nickel. So he's made a little magnet, and many shapes, cylinders. He's made ellipse. You can microfabricate simple disks. And the nice thing about microfabrication is almost everything's the same. In the case of the disk, we can get up to 90% iron, coat it with gold. All of the other particles are 40% iron oxide, so these are much more effective, these disks, than any of the particles. And the cool thing about shapes like cylinders and disks is you can calculate the magnetic field when you put them in a magnetic field, and they've been designed more or less to have a homogeneous volume, so their thickness and spacing is set so that even though when we think of iron oxide, we think about inhomogeneous fields, right in the center of the field is very homogeneous. And that's the point. That's what takes an incoherent field gradient and turns it coherent, making it homogeneous. And since these are magnetic in the field, they're shifted. And this shows a shift, expected, a theoretical shift of water that's sitting in the middle of the disk. And what's cool about this is that now the shift, it's not chemical shift, it's physical shift. It depends on the property of the device. So if the spacing gets a little further away, maybe you can see here compared to here where it gets closer, the peak will shift. And if you use less metal, make each of the disks thinner, again, you'll get a different shift. And we first made these, we couldn't find them in the proton bandwidth of our spectrometer, so we added deuterium to lower the gamma, so smaller shift, and we found it, and it would have been in the fluorine window, the original one we made. So the potential to shift is over all of magnetic resonance frequencies from very high to very low. So Gary likes to say we can make millions of these like RFID tags. In principle, nowhere near that. And so he took these three shapes, and you can see there's the red, and there's the G for green, and there's the blue. And so Gary likes to say, oh, we've added color now to these iron oxide particles. We've shifted the frequencies. And this is work that's been going on for a while. So now back to Paul. So now rather than an incoherent process, we have a coherent process. And basically, if we can put something between these disks and get them to change their geometry, they become sensors. Does the disk move? So here, if we could have that happen in response to something, it's really a beautiful sensor in the way Paul meant to build, I think. And Gary went and did that, and he made the post out of a polymer gel that expands and contracts based on pH. So that's the idea. Acidic close together, shift far from the water, basic further apart, shift closer to the water. So just to turn this idea into a dynamic sensor, and he succeeded. Here's the electron microscope that shows, again, the nickel disk with a hydrogel post. This is a gel that will contract and expand based on pH. These gels are amazing. As I said, Paul was quite fascinated by them. You can imprint molecular structure onto them, so they really can sense many, many, many things. And here's the first results. There's the shift at pH 5, the shift at pH, some correction for the water background there, and there you can see the shift with pH. And so again, a paper at 93, it's haunted me isn't the right word, it's inspired me for a long time, and I finally feel like we're maybe on track to realize that idea of Paul's. So let me conclude. I've shown you two things, the cell tracking, and that one I feel these cell-based therapies are going to explode, and that then tracking those cells will have to explode as well. And in particular, T-cell engineering for tumors looks remarkably promising, and stem cell therapies are also there. So I think the biology is going to get back to driving the contrast in this case. Of course, the challenge are all the other things we'd like to measure. Like Paul, I told you we love just manganese ion. But what we're missing is really a flexible contrast agent platform that's highly potent, highly responsive, and that can be used. And I'll just close. It turns out, and Joan can probably correct me, I'm probably wrong again, I think this is Paul's last paper. Published in 2010, came after the evolution paper. And here he is with these gels, microwave-assisted salt gel synthesis for molecular imprinting. So it's something about these gels I think is going to give us this platform. I'm not sure about the way we're doing it, or the way the Weisletter group's doing it, or the way the Jasanoff group, but I think Paul was right on in this thinking. And it's 1993, it starts 2003, 2000, it's only 20 years later, so it's still a ways to go, and that's clearly the kind of thinker Paul was 50 years down the road. So let me conclude from the last paragraph, back to Papa. From the last paragraph of that Nature paper is this statement: zomatographic techniques. I'm sad that we dropped it. It's nice. That's Greek, and Quo Vadis for me is Latin. Should find many useful applications in studies of the internal structures. All right, so that's MRI. We do that all the time. But then he's got this states and composition, spectroscopy, maybe deals with composition. We're waiting for that to fully flower. I don't know what he meant by states, but of microscopic objects he had in mind. And what I think it's clear, those microscopic objects are going to be inside of us. We're going to have microscopic resolution inside us. And so there's no doubt in my mind, in 1973, he invented MRI, he invented contrast agents, and he knew that what we call molecular imaging was on its way as well. So really a great honor for me to have had a chance to talk with you.
I know the hour is late, but before you go, please, Alan, accept this little plaque or diploma that says that we are recognizing your many achievements in imaging research. And this is in memory of the 2015 Lauterbur lecture, the first lecture.
Thank you very much.
Dedicated to MRI.
Thank you very
So
much.
thanks so much.
Oh, thank you.
Small memento is the history book of Case Western.
I started this already in your office, so this is great.
I'd like to recognize Joan, if you don't mind, because since we have, you love to write books, we have another book for you. So I'd love for to everybody recognize Ms. Lauterbur, who has provided also a scientist in her own right, who provided all this.